We move on now in Science 200 to talk about the origin of life. And we start with a concept called chemical evolution that will use a lot of the material that you have already seen in previous lessons and puts them together into a way of understanding how life arose on this planet. From then on, we will use these concepts to build upon our current ideas on how biodiversity, all the species around you, arose from very simple principles of evolution by natural selection. So we begin this lesson using this short video that was put together by people at Georgia Tech that sort of summarizes a lot of the concepts that we will discuss in more detail in the class slides. For now, let's pay attention to this and, as always, take good notes because it's important material that we will expand upon later.
Stated Clearly presents, Can Science Explain the Origin of Life? A full scientific explanation for the origin of life does not currently exist, but research groups around the globe are tackling the mystery. Here we'll take a look at a few of their discoveries. Darwin's theory of biological evolution tells us that all life on Earth may have originated from a single, relatively simple reproducing creature living in the distant past. This idea is based on many observations, one of which is that when living things reproduce, children are often born with random new traits. Those with negative new traits are less likely to survive and reproduce. Those with positive new traits are more likely to survive and pass those traits on to their children. Over multiple generations, positive traits build up, allowing relatively simple life forms to evolve into complex life forms and even split into multiple different species. A careful look at the fossil record overwhelmingly confirms Darwin's theory. Life forms get simpler and simpler as we go back in time. The record suggests that all life on Earth emerged from primitive cellular colonies, which first appeared roughly 3.5 billion years ago. But how did those first reproducing creatures come about? Even the simplest bacteria alive today are far too complex to have popped into existence in a single step. Furthermore, the first reproducing creature could not have developed through biological evolution because biological evolution requires reproduction in order to work. The origin of life needs its own explanation. The most promising idea right now, the one which is consistently leading scientists to new discoveries, is the idea that life emerged from chemistry. Chemistry is the study of matter, particularly how atoms interact and combine to form molecules, and how those molecules interact and combine to form larger systems and structures. Everything is made of chemicals. The air we breathe, the ground we walk on, the shoes on our feet, even the feet inside our shoes. That said, the chemistry of life is special in two very important ways. First, the chemistry of life is organized into metabolic pathways. If you look at the cells of any living thing, even a plant which you wouldn't normally think of as being very lively, you discover a world of activity. Chemical reactions occur in continuous orderly paths, each reaction sparking the next. Scientists call these strange reactions metabolic pathways. The second oddity of life's chemistry is that living cells are made of special molecules. Building blocks like amino acids and sugars, which we once thought could not be found outside of life. These building blocks combine to form highly complex and orderly structures like genes, proteins, and cell membranes. In the past, scientists had no idea what powered the strange activities of life and produced its unique chemicals. They concluded that life must be fueled by a mysterious energy they called the vital force. In 1828, a chemist by the name of Frederick Wöhler accidentally produced urea in his lab. Urea is one of the main components of urine, one of many chemicals that the scientists of his time thought could only be produced by the vital force acting inside of a living creature. The artificial production of urea suggested that life might not be powered by a mysterious force, but instead is powered by normal chemical reactions which could be studied and understood. Since that time, numerous molecules and cellular activities have been recreated in the lab, clearly demonstrating that life is powered by normal chemical reactions and suggesting that life may have emerged from non-living chemistry. Researchers have recently discovered that many of the building blocks of life, amino acids and sugars, exist inside of meteorites, rocks which have fallen to the Earth from outer space. This tells us that these special molecules are being produced spontaneously all throughout our solar system and may have been common on the ancient Earth. We've also discovered a process now referred to as chemical evolution. When simple molecules are left alone with an energy source, they interact with one another, often forming larger, more complex molecules as time goes on. Experiments in the lab have shown that chains of chemical reactions can develop, some of which resemble the metabolic pathways of life. Surprisingly, we've also found that certain molecules have the remarkable ability to self-assemble into complex, orderly structures. Some self-assemble into hollow spheres, almost identical to the membranes of modern living cells. Others self-assemble into long columns, remarkably similar to the strands of DNA found in life. Scientists still have many questions to answer about life's origins. How did different types of molecules first start working together, eventually producing the genetic code? What were the original reproducing creatures actually like? Were they similar to the cells we have today, or were they something much simpler? And finally, possibly the most interesting question of all, how likely is it that life has formed or is forming in other regions of our galaxy? So, back to the original question, can science explain the origin of life? Science has not explained the origin of life in full detail, but researchers are building a theory intended to one day describe, start to finish, how basic chemistry can give rise to living cells. I'm John Perry, and that's the origin of life, as we understand it so far, stated clearly.
So as you can see, we are not all in agreement, and of course there are some gaps in our knowledge, but that doesn't mean that we have not made important discoveries and reached significant advances on our understanding of how life originated. It has to be put in perspective though, like everything in this class, we start with a little survey of the history and the early thoughts, and we have to go back to some things that are really not science, because we've been asking and dealing with these questions for as long as there have been humans looking at the skies wondering the big question, where did we all come from? And of course, the oldest answer has always been something or someone, some supernatural being that we do not completely comprehend made it. And that falls within what we call special creation explanations, this idea that something supernatural explains life and its origins. It depends on what religion you're talking about in terms of the details, but the concept is the same. Whether it was made in one step out of nothingness, such as the case for Hindus and how Lord Brahma created the universe or the world, or whether it happens in steps, which is the case for Muslim tradition or Judeo-Christian tradition, the outcome is the same. An all-knowing being was behind this. Later on, people started questioning not the concept itself, but some of the assumptions of it. And people started talking, as the video points out, about things like vital force, an invisible force that would lead spontaneously to the formation of similar life forms. So all the dolphins would spawn from this life force that is the life force of a dolphin, and all plants will spawn from whatever that life force is that creates only and exclusively plants. But still, it is an esoteric, a metaphysical concept. Later on, people started talking about hierarchies in the way that life originated. And since we are the only living beings that suffer from ego, it was very hard for people to accept that the same processes that would lead to the formation of an earthworm would be behind the formation of humans. So people started classifying living beings as a hierarchy, and they talked about lower versus higher life forms. And the idea was that lower life forms like earthworms or maggots or things like that that people observe were the outcome of decaying processes like rotting meat or leaves in the yard breaking down over time, that these were actually spontaneously formed. So not necessarily talking about life forming from chemicals as we understand it now, but actually organic life coming from inorganic sources, because the concept was that these lower life forms could not possibly be forming from the same processes that lead to the formation of something as complex as a human being or a dog or a cat. This idea of spontaneous generation was disproven by experimentation, and it's one of the earliest examples of how a large, sort of all-encompassing theory has been dismissed by using some semblance of a scientific method. What Pasteur did is that he put pieces of meat inside jars and left some open and others closed, and then of course the ones that were open developed maggots because flies got into the jars, laid their eggs and all that. So this is one idea that was experimentally dismissed out of scientific progress. Now what we know now is that life originated in a slightly more complex way than that, and we still don't have all the pieces of the puzzle put together, but we are looking into some really interesting places to try to piece it all back. What we know about life on Earth is important because it informs us our ideas about whether life can exist somewhere else. And the video points out something like that. Now that we have the technology of actually detecting chemical compounds in objects that are away from the boundaries of Earth, we are coming to terms with the fact that these organic molecules may not be as rare as we thought that they were. Now, when it comes to life as we know it and as we define it, in the simplest, most basic terms, you need two things. We already established that we are organic life forms, and an organic molecule is one that has carbon attached to at least a hydrogen. And that means that in order to create a living being, you need a source of carbon. The other thing that we already established from the lesson on the characteristics of life is that there are energy transformations that happen all the time inside cells. So the other thing that you need is an energy source. So what we've come to realize by looking at the most bizarre, most extreme, most inhospitable environments on Earth is that life finds a way. So as long as you have a source of carbon and a source of energy, it doesn't matter how extreme temperatures, pH, salinity, pressure, or any other physical variable are, you will find some organism that has adapted to live under those conditions. These organisms that live on these very bizarre conditions, or conditions that we couldn't live under, are what we call extremophiles. And they include bacteria, for example. This Thermus aquaticus lives in Yellowstone National Park in areas where the magma that is underground heats the water and makes it boil. So these bacteria have evolved to live in boiling water, basically. If you look at the Atacama Desert, which is one of the driest areas on Earth, it's a desert in Chile. And if you were to take a geological sampling hammer, the ones that are kind of thin on the tip, and break open one of these rocks, what you would see is that underneath the surface there would be this sort of blackish greenish layer. And it turns out that these are living things. They are groups of bacteria and microbes that live underneath the surface of these rocks in these what we call consortia, these operating groups in which one microbe provides one thing and another one provides another, and they benefit from each other, and they form these layers inside rocks in one of the driest places on Earth. So life can exist under conditions in which we would not be able to as human beings. This also reinforces one concept. We call these extremophiles or extremophile organisms because they live under conditions that we as humans could not live in. But for an extremophile that lives in boiling water, our normal temperature would be extreme. So it's important that we keep this in mind, that we define extreme from our very own human experience and perspective. It may not be the same for the organisms that live there. For an organism that evolved to live under no oxygen, living in a simple oxygen atmosphere like we do, is extreme. So extreme is a relative term. To understand how life originated, we have to go back to a concept that we've already mentioned before, and that is that everything in the universe is bound by the laws of physics and chemistry, and that includes living beings. So, hypotheses about the early events of the origin of life have to be bound by the laws of chemistry and physics as we understand them. So this idea of chemical evolution is only a manifestation of these principles. We also have to keep in mind that we're trying to talk about the past, and we're trying to pierce into the deep past of the early stages of formation of the Earth, which means that conditions back then were really different than the conditions now. So we need to understand that we are reconstructing earlier stages of what the atmosphere looked like, what the surface of the Earth looked like, what environments look like, in order to really make full use of these chemical-physical principles. We know that the early atmosphere in the Earth, because of a number of techniques that we can use to trace this back, we know that the early atmosphere was very different, and that was mostly comprised by CO2 and other gases that were released by volcanoes. In other words, oxygen was very, very low in this early atmosphere, and that's really important because a lot of the chemical reactions that we're going to talk about that gave rise, or we believe gave rise to life, would not happen in the atmosphere that we have today. Another thing that happened is that rain eventually developed in the atmosphere, and it became part of what we call a hydrological cycle. So minerals and salts were eroded and dissolved out of the rocks that were forming in this early atmosphere and in the early seas, in the early bodies of water that were forming here. Now, a lot of the question, of course, is where does the water come from? And you already have partial answer to that. A lot of this water was probably brought in during the early stages of the creation of the solar system, in which there were a lot of things hitting the Earth. There were a lot of comets and meteorites that were landing on the Earth, and many of them had water ice. Now, some of it was also released as part of the volcanoes that we're talking about. So it was a mixture of origins. We know this because we can study now the composition of gases that come from volcanic eruptions. We also have meteorites that we have collected, and we have instruments that can go and measure the chemical composition of objects in space as well. We have made studies on ancient rocks that we have dated to millions and billions of years, and we know a little bit about how things are working in some of the planets around our solar neighborhood. We now even can talk about how things in planets away from the solar system are. So we've come a long way in trying to understand this. A new term that I'm giving you here in this slide is that of geochemical cycles, and as the word implies, these are transformations of matter, of chemicals that are on the rocks from one form to another, and these connect all these processes through the entire Earth. We will revisit this later on when we talk about ecosystems and eventually the biosphere as an interacting set of geochemical or biogeochemical cycles. But for now, understand that these geochemical cycles have to do with some of the concepts that we already established in terms of transformations of matter from one form to another on Earth. Small organic molecules that are those molecules of life that we know now, those building blocks that we already talked about, can be formed by non-living mechanisms. And the video puts that in perspective very well by mentioning the pioneering work of Wöhler, who was able to synthesize urea from inorganic sources. The reason why that is important, even though we take it for granted these days, is because back then it was believed that the molecules of life could only be formed by living beings, and that's how they got perpetuated. What Wöhler was able to do was to show that you could go from inorganic to organic through simple chemical reactions. But it wasn't until the Miller and Urey experiments, that there are several of these, that this actually started taking shape as part of a cohesive theory of the formation of life. What Stanley Miller and Harold Urey did was to try to reproduce the chemical reactions that would happen on an early Earth. And these were run between 1952 and 1953, and then reassessed by many scientists over the years. What they did was they came up with this contraption here that had what at the moment was believed what the original atmosphere of the Earth at one point in time would have looked like. So it had a lot of ammonia, and it had a lot of water and hydrogen gas and all that in a chamber. Now to that chamber, they inserted two electrodes that were supposed to simulate what would have happened with lightning in that early atmosphere. So this is your source of energy here. And periodically, they would spark a discharge, an electrical discharge in the atmosphere, and collect the outcome of whatever happened here in these glassware tubes. And they could actually take samples of that collected water with all the chemicals that were forming under these conditions. Remember, what they were trying to do is to simulate the early stages of the formation of organic molecules. And what they were able to show is that from very simple inorganic compounds, you could get actually 5 amino acids that we already knew about. So you could also get the formation of sugar, simple sugars, in this water that was accumulating here. Later on, people that did reanalysis using a similar machine were able to prove that you could get even more than that. For example, in 2008, a group of scientists reassessed this experiment. They redid a similar experiment in a more sophisticated and more accurate type of contraption, and the device yielded 22 amino acids. Remember, there's 20 common amino acids and 2 that are uncommon, so that was pretty close to what you find in life. Now, the 22 doesn't mean that they look exactly the same as the ones that you find in life, but many of them were exactly the same molecules. They found a couple that were not found in nature, but we knew that were chemically possible. Now, this looks like a very complicated slide, but I don't want you to get wrapped up in the details. I want you to remember the general concepts here. What Miller and Urey were able to prove is that you could get organic compounds from very simple chemical reactions that happen between inorganic components. And they grouped these into 2 main sets of reactions, the ones that were simple one-step reactions, and the ones that took the products of these reactions and led to more complex molecules here. So the chemicals in that artificial atmosphere that they created inside that piece of glassware that were exposed to a spark, an electrical spark imitating lightning, would combine in the presence of this energy source to form things like cyanide and formaldehyde, and those in turn would either spontaneously react to form more complex molecules, or they would react in the presence of that electrical charge to make more and more complex molecules. So the products of these reactions would lead to more reactions that can end up becoming amino acids in the end. So these compounds would react under these early Earth conditions to lead to some of the building blocks of life as we know it right now. That is amino acids and also single pentose, sugars like ribose, which is a 5-carbon sugar, and these could be explained by very simple sets of reactions. You don't need to know whether these are Sturcker synthesis reactions or what type of reactions there are. I include this information here for those of you who feel motivated to look more into these. And of course, every once in a while there may be an extra point here or there if you know these. But what I'm most interested in is that you know the basic concept here. And that is that simple reactions will lead to products that are organic, and those products themselves may combine into more reactions that would lead to increasingly more complex organic molecules. Of course, that's a far cry from creating something like a protein, which is comprised of long chains of amino acids that swirl and twist and turn into making tertiary or quaternary structures. So the question is, how do you end up from these very simple organic molecules with the complexity that you have today? And moreover, where does metabolism come from? How do these reactions get favored in the way that they get established as chains of reactions that happen and are codependent on each other? We know that all living cells carry out metabolism. That's a condition for life. We know that all cells are comprised by a boundary that is formed by a plasma membrane, which is made of phospholipids. All of them are of that type, at least all of the life forms that we know of so far. And we also know that all cells can only be alive because they have evolved the ability to replicate themselves. So these are 3 very important steps here: metabolism, the creation of a membrane or the existence of a membrane, and replication, reproduction. Where do these come from? Scientists have been preoccupied not only with explaining how these processes or mechanisms came to be, but also where they could have occurred for the first time. And they point out at 2 possible environments that were very common in the past, and they're still around, but they're not nearly as extensive any longer. One is what we call mudflats or tidal flats, and these are surface areas in which there is a lot of influence of shallow water, shallow ocean water, and there is an extensive layer of clay that covers the ground. This is what we know as mud. The other place that they point at is at the bottom of the oceans in areas where there's active spreading. So this is along the edges of spreading centers in those mid-ocean ridges. And these environments are what we call hydrothermal vents. So we will talk about both of these in detail. The first one are what we call tidal flats. And these are areas that you can see here. They're extensions on the surface where you have a lot of mud and silt and what we call clay particles. And these clay particles have a negative charge on the surface. So 2 things happen. Number 1, they tend to form hydrogen bonds with water molecules, and they also tend to form hydrogen bonds with each other. In these areas, sunlight was beating down constantly, and that could have provided the energy that is needed to bind simpler molecules into more complex ones. So in these tidal flats, which is the name of these environments, clays could have concentrated organic compounds by evaporation. So the compounds would be dissolved in water, and as water evaporates, the concentration of these organic compounds would increase because there's less water around. Under these conditions, then sunlight would act upon these molecules and provide the energy to bind them together into more and more complex, larger polymers, larger organic molecules. And there's some evidence that has been set forth from experimental work showing that if you make an artificial tidal flat in the lab and you have amino acids in a solution and you put these together under sunlight, the amino acids will eventually link together into small chains of amino acids that we call peptides. So the origin of these complex molecules that we know as proteins could happen from very simple principles of chemistry and physics spontaneously without the need of a living organism making them. In terms of hydrothermal vents, these, remember, are at the bottom of the ocean, and these scientists have pointed as the possible origin of other types of metabolic pathways, because if you focus and if you look under a microscope at the surface of these areas, the minerals that deposit when hydrothermal vents are active form these sort of honeycomb chambers. These are microscopic chambers, and these act as surfaces in which a number of organic compounds can bind to. Now, let's talk a little bit about what a hydrothermal vent is. This is an area, I already told you, these are in rifting zones, in areas at the bottom of the ocean where new crust is forming. So this is around the areas of rift valleys and those spreading centers in the mid-ocean ridges. Now, in these areas, the water, the seawater at the bottom, because there are all these cracks and breaks in the crust, the water sort of seeps in. But remember that these are areas that magma is pushing through. So the magma superheats the water up to 400 degrees Celsius in some cases, and the water sort of goes through the cracks, heats up, and shoots back out into the bottom of the ocean, and it forms these chimneys, depending on the minerals that are dissolved in the water. That when this happens, geologists call them either black smokers or white smokers. But for the most part, they carry a lot of minerals, particularly sulfides and other types of salts that the water carries as it gets heated and shot out back into the bottom of the ocean on top of the sediments. So these chimneys, basically, as the water, this is heated water, it's surrounded by really cold water. As this happens and these two water masses encounter each other, a lot of the minerals sort of precipitate and form these structures that look like columns that are continuously growing, as long as this process of water being heated and shut back out is going on. But what is exactly a hydrothermal vent and why is it important? Let's watch this video that has some actual live footage, and it talks a little bit about the discovery of these incredible ecosystems back in the 70s.
The Pacific Ocean, 250 miles off the coast of the Galapagos Islands. In 1977, a deep-sea submarine descended over 8,000 feet. Its mission? To investigate hydrothermal activity on the ocean floor. Marine explorer Robert Ballard was aboard. He detected a dramatic change in the ocean's temperature.
You know, we were not expecting what we found. We were expecting to find water, and we had a camera inside the vehicle, but it was just taking pictures, so we didn't know what we had till we came back. And we brought it up to the surface, and we processed the film, and we knew the spot where the temperature spike was, and it was like going to Disneyland. Probably one of the biggest biological discoveries ever made on Earth was made that day.
Ballard and his team were the first to see it, face to face. Hydrothermal vents. Immense chimney-like structures, several stories high, spewing hot water geysers, black with minerals and nutrients. The temperature around these deep-sea vents was a scorching 760 degrees Fahrenheit. And then an astonishing sight. Life, thriving without sunlight, a biological community never seen before. An exotic garden of marine life, species without eyes, others resembling Triassic-era fossils, over 200 million years old.
What we totally were blown away by were these giant tube worms, some of them 8, 9, 10 feet tall. And when you cut them, they bled human-like blood. I mean, when the submarine landed, there was a squish, and red blood came up around all the portholes. And that's how eerie it was. And then to find these extremely unusual creatures living in this oasis. It had no relationship to the normal life of the deep sea. And yet here they were living in this toxic water. But yet these creatures were thriving on it. And then when we dissected, I remember we took one of these clams, and we opened it up in the first place. Whew, as soon as we opened it up, it stunk.
It was full of hydrogen sulfide.
Horrible smell.
Rotten eggs.
Yeah. And we opened it up, and then we looked, and it didn't look. It looked like beef. It was red, bright red, and it didn't have an anatomy of a clam. It was like, what happened to the clam? Someone had taken over its body, and that something was a bacterium, a tiny bacterium that had figured out over eons of time how to duplicate photosynthesis in the dark chemically through a process we now call chemosynthesis. And that was the discovery, that there was another life system on Earth that did not go by the book that you and I read, that was not living off the energy of the sun, but was living off the energy of the Earth itself. And that really opens up the ballgame.
I'll say.
And these bacterium, we now think are the largest mass of living things on Earth.
That they're in the rocks, everywhere.
In the whole
under,
mountain range.
Under the ocean.
If you added up all the people and all the living things on land and add up all the creatures in the ocean and the few things in the sky, and you got a number, so many tons, there's more tons of biomass. We used to just think, you know, the insects ruled the Earth.
Yeah.
Wrong. These bacterium.
So where are these vents?
All over the Earth. They were, imagine a baseball with a seam on it. That seam begins beneath the polar ice caps, goes down through the Atlantic, into the Indian Ocean, across the Pacific. It runs around the Earth for 42,000 miles. It's the largest feature on Earth, the mid-ocean ridge, and it's underlain by literally tens of thousands of magma chambers.
Is that where life began, in the hot springs?
Well, that's what people are now thinking. The biology books that we were reading have now been thrown away.
Yeah.
And they're
when I
writing
grew
new
up,
ones.
everything, you had to be on the surface.
That's right. And you had this soup and the lightning bolt hit it and you formed all sorts of amino acids and things like that. But now what we think is that the hydrothermal vents may have been the site of life on Earth. It's also given us a new prospecting tool for searching for it elsewhere with our own solar system. We're now looking at a moon of Jupiter called Europa, which we think has an ocean. We think it has underwater volcanoes, and there should be life down there. The question is, how smart are their clams?
Did hydrothermal vents give birth to the first primitive microbes on Earth? Perhaps. But one thing is certain. Their discovery challenged long-held beliefs about the conditions necessary for creating life. And once life happened, there was no stopping it.
So what does that mean for the topic that we're discussing? How does that manifest itself into the first steps that led to the formation of life? Well, if you go into one of these hydrothermal vents and you take a sample and you look at it under the microscope, what you will see is that these little chambers act as surfaces that can allow organic molecules to sort of bind together. These iron sulfides that we talked about in the video that are mentioned here and that you can see here form this sort of honeycomb structure. And inside these honeycomb structures, molecules bind together. So these serve to concentrate organic molecules, and they act as what we call a cofactor for enzymes. So for proteins, a cofactor is a substance that is not organic, but that it enhances the ability of the enzyme to do its work. In other words, it makes it do its job better. Now, the iron sulfides can be cofactors of enzymes, and we have observed this in the lab. We also know that these areas, these surfaces, increase the active surface in which these reactions could happen, and that these reactions can happen one after another, coupled in a way that it almost resembles metabolism. So Iron sulfides in the chamber walls of these honeycombs that are, remember, these are microscopic, concentrate organic molecules and act as enzyme cofactors, both of which are key steps into understanding how metabolism may have originated. Now, this was a dramatic discovery, and the video sort of seems to suggest that scientists have completely dismissed that tidal flats may have had anything to do with life any longer, and everybody's focusing on hydrothermal vents. The reality is a little bit more complicated than that, and there's nothing to say that these two processes may not have been happening at the same time and may have led to different groups of living beings. Now that we know about the molecular and metabolic machinery, we need to ask questions about how the actual structures of the cell came to be. So where did cells actually come from? How did these molecules assemble into these structures that continue to perpetuate themselves? We know that vesicle-like structures that contain membranes can form by themselves when certain organic molecules are put under certain conditions, and you already saw this in one of the videos. But we also talked about the way that phospholipids behave according to their properties when you put them in water. So this should be reviewed in a sense. There is a term that we use to refer to this very early sort of in-between life and death structures that led to what we know now as actual cells, and the term is protocell. So a protocell is a membrane-bound sack of molecules, and it has the ability to do four things. First of all, it can capture energy. Number two, it can concentrate materials inside of it. Number three, it engages in metabolic transformation, so it is capable of doing metabolism. And finally, it can replicate itself, so it can make more. These are the basic conditions to see the first stages into what we call life. These occur because of properties of organic molecules when they interact together. So again, remember the concept of emergent properties. We have created things like this in the lab before. These are two pictures of outcomes of experiments. On the first one here, you see protein vesicles that are made by taking amino acids, put them in solution in certain alcohols, organic alcohols, and then heating the samples. These will self-assemble into these spheres when the mixture is in contact with water. Again, the temperature and the compounds involved here are meant to mimic the conditions that you would find in an early Earth. On the right here, this one is far more sophisticated and far more compelling as an argument. This is the outcome of vesicles that happen in what you would expect in a tidal flat. So this is the outcome of experiments that have been done with clays. And in this case, you had fatty acids that will self-assemble into molecules that will make the equivalent of a cell membrane, and those will even enclose RNA, which is these red spots here. These are RNA molecules that have been encased by these protocells, and these actually will perpetuate themselves. They will replicate. So these two are basically models of how these molecules, now that we know that they can form spontaneously, may continue assembling into the original cells. Both of these are examples of protocells in the sense that they are surrounded by membranes that are selectively permeable. And both of them have formed under conditions that simulate early Earth physical-chemical parameters. So there is something to this. It is possible that these molecules, these structures, can form from molecules that may have occurred in an early Earth under the conditions that we expect that early Earth had. How about the ability to build more based on a blueprint? People have proposed that the original information molecule was not DNA as it is now, that is actually a later development in the history of life. So people talk about an RNA-based system of inheritance that was the original one. In this case, this is what people call the RNA world, and this is at a time when RNA both had the ability to store genetic information, so the information to make more of these protocells, but also it had the ability to replicate itself. So it had both the ability to store information and code that information out. So in other words, it functioned like an enzyme in synthesis of new molecules. The evidence for this came with the discovery not too long ago, only a few decades ago, of what we call ribozymes. Ribozymes are pieces of RNA that have the ability to self-replicate, so they have information that they can use to make more of themselves. They can copy themselves into more molecules of the same kind. So these ribozymes lend a lot of support to the possibility that these were the original blueprints, and then something changed in the conditions that led to the formation and the evolution of DNA later on. What could have made that happen? Well, the one hypothesis is that DNA arose as a function of stability. DNA has two, as you know, it has two strands those two strands are bound by hydrogen bonds, and it makes that sort of spiral staircase type of structure. This structure is far more stable and resistant to breaking down than these ribozymes that we talked about, or any RNA, because RNAs are single strand. There are no double-stranded RNAs. So DNA may have been the molecule that survived as conditions change in this early Earth. And when we talk about conditions changing, we're not talking only about temperature, we're also talking about the composition of the atmosphere, which, as you will see, is a very important component of understanding this whole bigger picture. Now, DNA also allowed, as a consequence of being more stable, for larger and more stable genomes to evolve. In other words, you could store more information in these molecules. Hypothesis number 2, which is not necessarily mutually exclusive, is that DNA conferred resistance to early RNA viruses or any other kinds of molecules that would try to use the existing RNA to replicate itself or to consume it. So this idea is that DNA actually, by being double-stranded, conferred resistance to possible attackers, to possible viruses that would be attacking these early protocells. This is, of course, a temporary advantage only because eventually, as you know, viruses evolved to exploit DNA also to make more of themselves. But at least in this early evolutionary stage, this could have been an innovation, an early step into bigger, sturdier, more stable genomes. So with what we know, we can put together a broader picture of the steps that led to the formation of those first living cells. We know that in all living cells, proteins are important to have metabolism happen because enzymes are mostly proteins, but they're not the only ones. We also know that all cells have a plasma membrane that encloses them and provides a boundary, and we know that DNA now is the molecule that has that blueprint, so it's a molecule of inheritance. And we know that throughout history, over the decades, a number of laboratory experiments and field observations have provided different pieces of the puzzle. They have provided insights into how these cell components have assembled and what processes may have led to their original formation. So where we are right now is that we've gone from a completely abiotic world with non-organic compounds that will start assembling and form organic compounds. This would lead to the formation of the original lipids, carbohydrates, amino acids, and so forth, early proteins, small peptides, nucleotides, and all these would assemble through different processes into that original RNA world, and eventually the evolution of DNA simultaneously, while all these other compounds were sort of forming their own spherical structure spontaneously given the changing conditions of the early atmosphere. Now these two are happening at the same time, and eventually through a number of coincidences, accidents, and changes in conditions in physical chemical parameters, you would end up with spheres that would contain some of this DNA or RNA template. These would lead to the formation of more organized structures and really replicable metabolism because it was encoded in the information in those protocells. Eventually, the membrane-bound protocells emerge as fairly well-organized self-replicating units, and the first living organisms are here. That only takes us to prokaryotes. So all we've done is explain how the first prokaryotes form. In the next class, we're going to go a few steps beyond and talk about how you end up from prokaryotes to really complex eukaryotic cells, which are the ones that comprise your body and mind and most of the organisms that you see around you. So we're going to leave it here, and remember that the key steps is understanding how you go from an inorganic world to a prokaryote. In the next class, we're going to focus on how you go from prokaryotes to eukaryotes through evolutionary time, and also we will talk about how we classify living organisms according to the modern system of taxonomy. So we'll leave it here, and I'll see you in class.